So thank you for being with us today. I'm happy to see all of you here online for this talk of Professor Kathryn Hess Bellwald. Kathryn is full professor and head of the laboratory for topology and neuroscience here at EPFL. Her talk is entitled "A Case for Diversifying Your Mathematical Toolbox." To give you an idea of what to expect, the talk will last for approximately 40-45 minutes, and we have time for questions at the end. Kathryn agreed to take your questions in line, so if you have any burning questions, maybe put them in the chat or raise your hand that we see them. Otherwise, put them in the chat, and we try to address them after the talk. Before we dive into the presentation, I would like to thank Kathryn for availability this afternoon, and of course, the entire CIS team for organizing this event again. Kathryn, the screen is all yours.
Thank you very much. It's a pleasure to be here, be online like this, and to see everyone. So yes, so I decided to, since this is supposed to be a very interdisciplinary, transdisciplinary context for giving a talk, I thought I would talk about different ways that one can apply mathematics, unusual sorts of mathematics, perhaps, the sorts of mathematics people aren't used to applying necessarily in a range of areas, well, most of them related to life sciences for what I'll be presenting today, but where you think, oh, wow, I didn't know that you could apply that kind of mathematics to this area. So let's see. I am an algebraic topologist by training. So this is, if I were giving an ordinary math lecture, after 15 or 20 minutes, the board might look something like this. And you'd think, well, how could mathematics that is described with these kind of symbols be useful for sort of anything related to, I don't know, robotics or machine learning or the life sciences? But it turns out that maybe not exactly this kind of algebraic topology, but certain flavors of algebraic topology are indeed very useful, as other areas of pure mathematics that haven't been much used until recently have proven in more recent times to be useful, such as the role that differential geometry is playing in machine learning these days, turning out to be extremely important. So this talk is meant to be kind of a pitch for expanding one's mathematical horizons, reaching out to mathematicians who may be able to help you with a wide range of problems, and illustrating this by talking about how my collaborators and I have applied the tools of topology to solve a number of different problems in the life sciences, but not only in the life sciences. So I'm going to start by talking about what topology is, since not everybody knows. It's not a subject that is required of most engineers at EPFL or most scientists, certainly not something that's taught in the gymnase. So let's start by explaining what this field of mathematics is. So here we have a little word cloud that puts topology in context. You can think of it as it's sort of in the same family of mathematics as algebra and geometry, but it's sort of has some discrete type aspects and some continuous type aspects. It's like geometry, but without any notion of distance or metric or angles, things like this. People call it rubber geometry, because what you're interested in studying is not sort of detailed geometry of shapes, but sort of in a more general form. And so here you see a bunch of sort of key words that come up when talking about topology. Now, so what is topology, maybe slightly more precisely? So it's the mathematics of shape. Now, I imagine many people have heard the joke about topologists not being able to tell the difference between a donut and a coffee cup, because one can make a continuous deformation of a coffee cup to turn into a donut, and vice versa. So what topologists are interested in is what is preserved when you perform these sort of continuous deformations of shapes. What is invariant under these kinds of transformations? Topology is also the mathematics of connectivity, not just are two things really connected to each other, such as by a directed path in this graph, but in how many ways are they connected? And what are the relationships between the different ways they're connected? And what are the relationships between those relationships? And so on. So really connectivity in all of its deep glory. And it's also, and this is perhaps most important for a lot of the applications that I have in mind, the mathematics of the emergence of global structure from local constraints. So the idea is that if you know sort of locally, you know some sort of shape, and you know locally various properties of that shape, what can you deduce about what sort of global structure might emerge from those local conditions? Topology is one kind of mathematics that is very good for studying those sorts of problems. Now, one of the most successful applications of topology in the last 10 to 15 years or so has been to data analysis. So it's a new sort of branch of mathematics at the interface of topology and data science called topological data analysis, in which one studies the shape of a data set. So this is usually some sort of high dimensional data set, usually a data set that has a lot of points in it. And you find a way of associating to this data set some sort of signature. And there are various ways one can compute this signature that encodes really the multi-scale structure of the data set. So you're looking at it, it takes into account sort of simultaneously the shape and the metric information about the data set. And by doing so, one can reveal important relations among these data points and use machine learning then to provide a fine analysis that leads to various classification problems or proposing clusterings and so on. So this has been a very successful new area of mathematics. One can also apply topology to analyzing different types of networks. So you can have a network that is either directed or undirected with weights on the edges or not, and so on. And one way to approach studying networks like this in a way that allows one to apply the tools of topology is to decide, well, what are really the significant subnetworks of your network? One might call them the motifs or the graphlets that you're interested in. And you want to focus on certain specific kinds of networks. So the ones that come up a lot have come up in our work in neuroscience, for example, are little subnetworks of this particular form, which are called directed simplices, and one can continue building subnetworks of this type. The point being that all of these little subnetworks here are of the form that you have, they are acyclic, there are no cycles in these graphs, there's a specific source, there's a specific target. And if you remove one of the nodes and the associated edges, you obtain a directed simplex that is one dimension lower. So deciding which of these significant subnetworks you want to study based on the particular context, based on why you're studying the network, what it arises from, gives you a new tool to study them. So one thing you can do that is more just directly combinatorics or graph theory is to study the numbers of the different types of the significant set networks that you have in a given network. And this is you giving sort of local information about the network. But often what you'd really like to know is to have some sort of global information. So you can say something maybe about the dynamics on the network or something like this. And a good way to study that is to quantify how these particular kinds of subnetworks overlap in your big network, and that gives you interesting global information. So now I'm going to focus on the specific application of topology to give you an example of just how powerful these methods can be, even with a relatively simplistic approach from a certain point of view. So the projects I'm going to be talking about are all led by Lida Kanari from the Blue Brain Project. And it's my pleasure to present our joint work here, that we've been working together on this for the last 7 years, I guess. So we're going to talk about how we can use topology to help us classify and better understand neuron morphologies, the shapes of neurons. So what you see in this slide here is a visualization of what are called the pyramidal cells from the cortex, from the higher part of the brain of a rat. And you see that they come in a wide variety of shapes and sizes and sort of how branchy they are and how curved the branches are and so on. And the reason why one is interested in classifying neurons in terms of their morphology is that shape has tremendous impact on the function of a neuron. So if you want to understand a neuron's function, you really need to understand a lot about its shape. And classifying neurons by their shape, by their morphology, is very useful from that point of view. So how can one go about doing this? Well, let's go look at a simple case where we just have two different neurons. Now, the one on the left, the one on the right, you say, well, they don't really look the same, but can I quantify what the difference is? Can I really make explicit how these two neuromorphologies are different and to what extent? So the method that we developed is called the TMD, the topological morphology descriptor. And let me see if I have a pointer. Yes, just a second. There we go. Okay. So I'm going to explain, it's a really quite a simple algorithm, and it's an example of something that is called persistent homology in dimension 0, but we can just do this quite simply. So the idea is that we're going to take this neuron here, which is represented as a tree. So of course, we're looking at a slide, so it looks like it's embedded in 2 dimensions, but this is actually works in 3 dimensions, if you'd like. So we have this combinatorial tree embedded in 3 space. And what we do is we have an algorithm for breaking this tree up into its branches, associating to that what is called a barcode. And that provides a very simple topological signature that it captures information not only about the underlying sort of combinatorial tree structure, but how it's embedded in space. So the idea is you start with the leaf of the tree that's the furthest out. You go to the first bifurcation point that you reach, and then you say, okay, we're looking at trees that only have bifurcations at each vertex, because that's what shows up in neurons. And you say, I'm going to break off the branch with the leaf that's the closest to the root, which would be represented by the cell body of the neuron. We'll break this off and record that branch as an interval, which is this interval right here, where you're just recording the distance of these 2 vertices from the root. And you continue on to the next vertex, and here you're just comparing this leaf here to this leaf here, which is left. This one is closer to the root, so we break off this branch, record it as a bar here, and we continue on. And if you go through this whole process, then in the end, you're going to have broken up your tree into a number of branches, where at each bifurcation point, you break off the shorter branch, the one with the leaf closer to the original root. And you obtain what is called a barcode for obvious reasons. And a barcode is just a collection of intervals in the real line. And you can alternatively record a collection of intervals in the real line as a collection of points in the upper right-hand quadrant of the plane, where you record the start and end points of each of these intervals. And so this is a very simple way of recording how this tree is embedded in space. And it seems like almost surprising that something like that would be able to tell you much about, be particularly useful for classification. So, let's do this again. Okay, so when you're doing this, it's possible that you could be making that when you're looking at a tree like this, you would like this process to give you something that is stable with respect to small errors, because when you're looking at these neurons, where are they coming from? They're coming from what are called digital reconstructions of actual biological neurons. So one takes a slice of actual brain tissue and looks it under a microscope, and then some poor PhD student or postdoc records the coordinates of the 3D coordinates of lots of points on an individual neuron. And that's the way the shape of the neuron is recorded. And of course, when doing those measurements, it could be that you make some kind of mistake. So you could, for example, have something like this, where all of the measurements are slightly off. You got all the branching structure right, but your measurements are not quite right. Or it could be that, after many hours of looking through a microscope, you missed seeing certain branches, or you added a few extra branches and so on, but just small errors like this. And what you'd like would be for this topological signature, the TMD, this barcode, not to be too sensitive to those kinds of errors. And indeed, we're able to prove as a mathematical theorem that this TMD is actually stable with respect to these kinds of small errors of reconstruction. So you have a desired stability. This is a worthwhile invariant with that. So there are various ways in which one can represent this data. As I said, you can start with a barcode like this, which has the more fancy name of a persistence barcode, where you've recorded each of the branches here as an interval in the real line. Or you can alternatively record each of those as a point in the plane, where you're just recording the starting and end points of each interval, because that determines the interval. These 2 representations are equivalent. Or if you want to actually be able to apply the tools of machine learning, this sort of output, having a collection of points in the plane or having a collection of intervals like this is something which is harder to input into machine learning. So you can do classification or something like this. So the solution, one possible solution to this, is to use what's called a persistence image, where what you do is you associate a Gaussian kernel to each of these points in the plane. You sum up the Gaussian kernels, and that gives you a function on the upper right-hand plane. Now that you have a function, that's something that's an object with which you can do statistics. You can take averages, you can take standard deviations, whatever. You can actually perform statistics when you have something like a persistence image like this. So a sum of Gaussian kernels around each of these points. And that's exactly the kind of input that we use when we apply this tool to doing various classification problems. So for example, here's really a toy example. We're looking at a comparison of neurons between different species. So here we have cat, dragonfly, fruit fly, mouse, and rat. And if we just look at them as humans, we can say, well, of course I can see, even though I know nothing about neurons, I can see quite a clear difference between the kinds of neurons one sees in cats or dragonflies or fruit flies or whatever. But the point is, you would like a computer to be able to do this work for you, so you don't have to spend your time looking at all of these digital reconstructions of neurons. So what we did is take populations of neurons from these 5 species, compute the associated barcodes, these topological morphology descriptors, or equivalently, or the associated persistence images. And then in this column D here, what we have is the average persistence image for the whole population of, in this case, cat neurons, dragonfly neurons, fruit fly neurons, and so on. And all of a sudden, we have a nice visual summary that's even more straightforward, I think, than what we see here, that enables us to compare these different types of neurons coming from different species. And indeed, when we use these persistence images with very straightforward machine learning, quickly with just a random forest, then we ended up with very high accuracy for classification of the four types. So that was just a test case to see whether this would actually work. So going back to these 2 neurons that I started with, one, we wanted to try to see, can we really quantify what the difference between them is? Well, so we take this guy, we had calculated its associated barcode, associated persistence image now. And the way to read these persistence images, so this one is associated to this neuron, is to say, this hotspot here corresponds to the fact that there's a lot of branching going on around the cell body, around the soma here. And this little hotspot here corresponds to the fact that we have these extra branches up here in the tuft, as they call it. On the other hand, if we look at this one, yeah, so it has a hotspot, a lot of branching structure around the soma, but there isn't much branching up here, and that's represented by the fact that we don't see anything here in this persistence image. We take the difference between the two, and now we can really see, so this square here is computed by taking the difference between these 2 functions. We can really see where these hotspots are, and we can now quantify, say precisely what the difference is between these 2 neurons. And again, this is something we could learn to do by eye, or some people can do it extremely well by eye, but this way we can automate and objectify the construction. So precisely, we use this in the context of looking at those rat pyramidal cells, like that first image I showed at the beginning of this section, where here we see all of the different kinds of rat neurons. So they're classified by at what depth in the cortex the soma, the cell body is. So the cortex is divided to 6 different layers. And then the neurons are classified by the layer in which the cell bodies are. So here, layer 2, 3, 4, 5, and 6. And then here we have representative neurons of various types. And what we did is see whether there was an objective way to classify these neurons according to their morphologies, and see how well it matched with what the expert subjective classifications were. And it turned out to be a good match. There was some clarification that actually came from looking at this method, doing the subjective classification using the TMD. And so it was another nice proof of concept that this was a good way of having an objective classification of different kinds of neurons according to their shape. Finally, a really interesting example came from looking at human pyramidal cells. So there are many fewer examples one has of human pyramidal cells that one can study in the lab. But the team at Hebrew University, the lab led by Idan Segev, had many examples of these human pyramidal cells. And they had the intuition that there should be a way of classifying them based on their function, but we're not really able to do so until Lida Kanari, who's worked on this project, decided to apply the TMD to this. Now here, the thing is that unlike the previous case where there was known classification known to expert, there was no known classification here. And so there was no way to train or test. It was just unsupervised learning that went on. And when we applied unsupervised learning in this case, we end up with a very nice way of defining two quite different classes of neurons. Some that are called slim tufted and others called profuse tufted. And what was really interesting is it turned out that once you had this classification based on morphology, it turned out that they had actually, this was underlying a classification based on function. That in fact, the function of these profuse tufted neurons was quite distinct from that of the slim tufted neurons in terms of their electrical behavior. So that was a very interesting application of this method, I believe. Okay, so now I want to talk about how we can sort of run this process backwards and start with one of these topological signatures, like a barcode or something like that, and then generate synthetic neurons, that is to say, neurons that are not actually biological, from actual digital reconstructions that have believable biological shapes and believable biological function. So this is an article that will come out a little later this year. So this is really an inverse problem, right? So what I was just talking about is how you start with a neuron, and you can break it up by breaking up all the bars, all of the branches into bars, and associate to it a barcode. And we'd like to just kind of go back. If I have a barcode like this, is there a way to build something that really looks like a neuron? Could I know where this is coming from? Now, it seems like we lose an incredible amount of information when we go this way, which indeed we do, but we'd like to try to go back. Why would you want to do that other than it's an interesting mathematical problem? Well, Lida's main motivation for working on this had to do with the blue brain microcircuits, which is part of the blue brain project. And for building their digital reconstructions of part of the rat brain or the mouse brain, they need large populations of neurons to build this. So in this case of the blue brain microcircuit, the original from 2015, there were roughly 31,000 neurons that populated it. And they can't all be copies of the same neuron. There needs to be the kind of diversity that one finds out in real life. So you need to either take the digital reconstructions of actual biological neurons that exist and somehow perturb them so that there's some diversity, or try to synthesize some that actually look like what you want. So the synthesis algorithm that we developed works as follows. So what you want to do is actually build these neurons as using a kind of random walk. So once you have your cell body, you're going to have the neurites, so these branching structures, in this case, the dendrites, which are actually gathering information from elsewhere and transmitting it to the soma. If you look at how these things are built, you're going to build this as kind of as a random walk, where at each step in the random walk, you either continue, bifurcate, or terminate. And as the dendrite elongates, there are various elements you take into account. There's a certain degree of randomness, a degree of memory of where you've been, a degree of targeting to get to a particular place. The way barcodes, these topological signatures come into it is you'd say, okay, I'm going to start with one of these barcodes coming from an actual neuron, and I'm going to grow this neuron step by step. And according to have an exponential probability of bifurcating as the distance from soma reaches the coordinate of this endpoint here, your chance of bifurcating increases exponentially according to some parameter lambda. And similarly, as you approach the other end of the bar, there's an exponentially increasing probability that you will actually terminate. So there's a stochastic element built into this synthesis process. And it turns out that there was this parameter lambda that comes in, and it turns out the parameter lambda that produces the biologically sort of best match and also most diverse population, the sort of a Goldilocks value of lambda is 1, where this is measured in units having to do with the step size of the random walk. But in that, you end up with a population of neurons that looks close enough to the neurons you started with, but they still have some diversity, which is part of the point. So this is just an illustration of what you get when you apply this process to various families of neurons. So here we have neurons that are either these pyramidal excitatory cells or what are called inhibitory cells in the various layers. And each of these illustrations, you see the blue neurons are actual digital construction of biological neurons. And the red ones are ones that have been synthesized according to this algorithm I just talked about. And just, sort of eyeballing it, they seem to be similar. Of course, we're scientists, and that's not enough for us. That's just a pretty picture. We want some actual, deeper statistical analysis of how close the match is. And here's one example of this statistical analysis. So what we did was start with a whole collection, a whole population of layer 5 pyramidal cells, or these digital reconstructions of actual biological neurons. And from those actual biological neurons, then looked at their associated topological signatures, these barcodes. And from those barcodes, we then generated a whole family of synthetic neurons, okay? And then the idea was to say, all right, there are various important morphological features of neurons, such as the number of sections, the number of terminations, the number of bifurcations, and so on, and looked at the distribution of these values over the entire population, whether it was this one or this one. And they did a violin plot to compare the distributions for the two populations. And again, this is more of a visual comparison. We can actually make this quantitative and numerical, but we see that our violins are pretty symmetric. So we've done quite a good job of matching the distributions of these various morphological features in the actual biological population as compared to the synthesized population. So it's a good match. We did a similar analysis. So you can take these synthesized neurons and actually innervate them in a way. You can actually give them a function and see whether they function similarly to actual biological neurons. And there as well, there was a very good match. So it's not just the shape that's correct. The shape actually, in terms of the influence of the shape on the function of the neuron, is also correct. One more remark about this is that the advantage of starting with using the barcodes to help you do this synthesis is that you're coupling the probabilities of bifurcation and termination as a function of these bars. And what this illustration shows is the following. So here we're looking at sort of these average persistence images, and on the projections here, they're the marginal distributions of the bifurcations and terminations. And this is what these biological reconstructions look like. When we do the synthesized cells, we see very similar pictures of the average persistence image and the marginal probabilities. Whereas if we uncouple the probabilities of bifurcating and terminating, so we have the same marginal probabilities, but we don't use any relationship between these probabilities of bifurcation and termination, we end up with a very weird collection of nothing that don't look like neurons, and the average persistence image doesn't look like what it should. So it's really important to use these bars to couple the bifurcation and termination. So these sorts of projects having to do with both classification and synthesis have been applied to other sorts of cells one finds in the brain, the glia, the astrocytes, microglia, and oligodendrocytes. So particularly in the recent paper by Interspecies, use this to synthesize astrocytes when making a model of the neurovascular vasculature in the brain, in the microcircuit. And in a recent preprint with colleagues from IST Austria, we used these methods for studying microglia in mice and ending up with interesting classifications in that case. We'd like to use this for application to doing comparisons between different brain areas and different species, the neurons one finds in different species. I would also like to improve the machine learning pipeline that we've used to this point, maybe involving GNNs or other more sophisticated tools. There's a team within Blue Brain that has worked on this. Okay, moving on. Another way in which we've applied topology in the context of neuroscience was in a paper that was written with people from the Blue Brain project back in 2017, where we applied topology to analyzing the blue brain reconstruction of part of the brain of a rat. So in this case, what we're studying is the following. It's a model built by the blue brain, a digital reconstruction of part of the somatosensory cortex of a 14-day-old rat. So this is I won't go into the details of how we built this model. There's a huge amount of work that went into it. But in the end, you have a working model of part of the brain with 31,000 neurons forming 8 million connections. So this is a very big graph. And you can simulate both spontaneous and evoked activity within this network. So when you think about two neurons, how are they actually communicating with each other? So here in this beautiful visualization from Blue Brain, we see a couple of neurons. Here are their cell bodies and these very, very complex branching structures that are their dendrites and axons. And these points that are lit up here correspond to what are called synapses. These are connection points between dendrites and axons, where the axon of one neuron reaches out toward the dendrites of another, and where neurotransmitters can transmit signal from one neuron to another. And the thing about these chemical synapses is that they really are connections with direction. There's a real sense of direction to the flow of information. And so it makes sense to represent a circuit like this, if you don't represent it mathematically, as a directed network or a directed graph. And so we can apply some of these tools that I talked about a little while ago for analyzing this. So here's an example of a very small directed network, but much smaller than 31,000 nodes and 8 million edges. And you think, well, how could I even begin to sort of quantify and describe carefully the structure of this directed network? And this is where these directed simplices come into play. You could say, well, how many of these directed simplices do I have as subnetworks of this network? Just count them from the various dimensions, and you can say, well, how do they actually fit together? What sort of more complex structures do they build? And it turns out that this sort of simplistic approach helps us to distinguish, to say interesting things about this blue brain microcircuit. As one simple example of what you did, you can sort of use it, to quantify to what extent this network is not random. So in this case, what we're looking at here is simplex dimension. So these are the number of nodes in one of these directed simplices compared to the number of these. And he compared the actual biological, this blue brain reaction, to something that takes into account less of the actual biology, to just a random network. And what you see is that the number of these simplices is much greater in the actual blue brain instruction than in a network that doesn't take biology into account quite to the same extent, or that it's even just a random graph. And indeed, there are even simplices up to dimension 7, which means you had up to 8 neurons were all connected in a directed way like this. And so this was it's one way of quantifying the degree of organization, because creating such high dimensional structures is not something that's going to happen randomly. And you can say, okay, but right, they give us a way of distinguishing these things, but do they have any functional role? And so just to illustrate, one of the experiments that we did was to say, okay, do these directed simplices have any particular function? So what we did was to say, we look at all pairs of connected neurons in the network and look at the correlation in their firing behavior. So you're looking at some sort of Pearson correlation in the what are called the spike trace of these neurons. And you want to say, okay, so to what extent does being part of a simplex have an effect on this? And so what's called dimension here on the x-axis is the dimension of the largest simplex to which a connection belongs. So I look at a particular connection, I look at all the different simplexes to which it belongs, and I say, what is the highest dimension of those simplices? And that's what I'm measuring here with dimension. Now, if we do the average of this correlation over the entire network, we end up with something like 0.3. However, if we look at connections that belong to simplices of high dimension, their correlation in firing behavior is much higher. And also it depends on where exactly within the simplex the edge is. So here, if we have the edge that is towards the end, at the very end of the simplex, at the ultimate connection in the simplex, you have the highest degree of correlation. And if you have something that is at the very beginning, you have sort of the lowest degree of correlation. Another interesting thing to note is that if you think about what it means for dimension 1 here, for that dimension to be 1, that means that that connection is not part of any simplex. And in that case, the correlation is much lower. So it's not enough for two neurons to be connected. They have to be connected and part of something larger. So that was an indication that the simplices actually play a functional role. And we will go into this in more detail as well. But this is sort of the entry point. Some more applications of topology, just to show that it's a tool that's useful across a number of domains. Again, still within neuroscience in this case. We're able to use these methods of topological data analysis to classify neural dynamics. So looking at a network of artificial neurons, you can simulate dynamics in this network of neurons, and you could use the methods of topological data analysis to be able to classify these neural dynamics with very, very high accuracy. So that was really nice to see. In a very different way, we used methods of topological data analysis, particularly tools called MAPPER, to develop a topological biomarker for long-term schizophrenia. So this was just looking at clinical data, actually, from schizophrenia patients, and using that clinical data, applying topological data analysis to that clinical data in order to predict how well the schizophrenia patients will be doing 2 or 3 years down the road. And again, the topology helped us to distinguish 3 different classes of these schizophrenia patients, those who would be doing the best in 2 or 3 years to those who were still going to be having a lot of trouble. More recently, in work with the lab of Brian McCabe, we've used topological data analysis to validate the existence of a sexual dimorphism in the brain structure of Drosophila larvae. So in Brian's lab, we do amazing things with being able to find the precise positions of neurons in these Drosophila larvae, and then looking at those positions as giving us a point cloud, we were able then to apply methods of topological data analysis and describe really, make evident the existence of a real sexual dimorphism that went beyond sort of how many neurons, but actually much more complex, like what was the shape of the point cloud given by the positions of these neurons. And then on a completely different note, in a collaboration with Baron Smit, chemical engineer based in Sion, the Sion campus of EPFL, we use the methods of topological data analysis as a way to develop a high-throughput screening for nanoporous materials. So you want to you have these nanoporous materials, and you'd like to find out which ones have high performance properties when it comes to storing CO2 or methane or something like this. And doing so by doing molecular simulations the results are very good, but it's very expensive. And the idea was to say, what if we just choose a few of these structures that we know have excellent performance properties, and then we look for structures that have similar shapes, similar to, topological structures that we could measure using tools of topological data analysis. And this turned out to work extremely well for helping us figure out, okay, these structures should have high performance properties. Let's focus on those. Let's do the molecular simulations for those. Let's maybe even synthesize those and see whether they actually turn out to be good. And it worked indeed very well. So that is all the different applications of topology that I share with you today. I'd like to thank the Fonds National Interspecies and the Blue Brain Project for having funded a lot of our research. And I'd like to thank my collaborators within the Laboratory for Topology and Neuroscience, a great team to work with, cover a wide range, and it's great to collaborate with them and with all the other people who share in this project. I really enjoy these sorts of interdisciplinary collaborations, and I'm very open to questions or to contact from people who are interested in seeing whether these methods could be useful for them. Thank you very much.
Thank you very much. Also the advantage of being physically somewhere for a talk, you can applaud and thank the speaker for his or her talk. Are there any questions in the audience? Don't be shy, just raise your hand, unmute yourself. I suppose people can contact you, right, if there are any questions coming up.
For sure. No, I mean, I have a great team of enthusiastic young people who would, you know, they're always really excited about new applications, new projects, and you know, I mean, one thing I didn't talk about was because it's not my work, it's the work of my postdocs. They've been working a lot on integrating some of these TDA methods and machine learning. There's some really cool work coming out there as well. I have another student who's working on developing really interesting topological methods for analyzing single cell data and looking at this RNA velocity and stuff like this. So it's really cool to see that, you know, these sorts of techniques can be applied like all over the place. And you know, I'm sure there are plenty of possible applications that I haven't thought of and that I'd love to hear about. But also I would like to encourage people to reach out also to other mathematicians. You know, it's like those who are working in machine learning, maybe it's time to have a conversation with the differential geometers. Or, you know, there's apparently this really interesting work they're relating algebraic geometry and things having to do with viruses and stuff like this. So I think that it's important to for everybody to be open to these kinds of really cross-disciplinary discussions.
Super interesting. Thank you so much once again.
You're welcome.
Then maybe if there are no other questions, then let me say some words of closing for this seminar, please. So thank you to you, of course, Kathryn, for your talk today. It's really interesting. Thanks to our audience for being with us. If you're interested, maybe we start with this Wednesday, the 2nd. We have our CIS RIKEN talk coming up with Professor Michael Unser. That's a seminar series we had the opportunity to launch together with our partners at RIKEN AIP, which features either colleagues from EPFL or colleagues from RIKEN AIP talking about the work. And then on the 7th of March, we are looking forward to welcome Professor Jean-Philippe Thiran, who is going to speak at our next CIS Get to Know Your Neighbor seminar. Thank you so much for your time this afternoon, and I hope to welcome you. And it's very important to say the next seminars are all hybrids. So don't hesitate to come to INF 328 and enjoy the talk and exchange with the speakers in person. Thank you. Stay safe and goodbye.
Thank you, Jan. Thanks, everybody.
Bye, Kathryn. Bye.